Hello friends, welcome to the microbial cell culture and its application lecture number 7th, and this is the last lecture for this unit. So far we have seen that how the microbial cell culture, microbial or microorganisms are cultured, what kind of substrates they need, what is the shake flask, what are the shaking water baths or shakers that are available, how you can do that, what are the reactors, what are the controllers in the reactor, how to scale up. Then we also had look at if the product is produced, can we downstream it, what are the implications of the different downstream processing, what is the cost implication in that. And then we had a look in the last lecture the strain selections and strain improvement, strain preservation. So, today we will just look at their applications. So, this we also had a look earlier as well, but this is just to recap and finalize what we have seen so far. So, for microbial culture technology, one of the application is whole microbial cells, and this is for food and vaccine. So, one of the most common example is algal based biomass. Spirulina for example is used a lot, is a very good nutritive food product, and that is basically being used. Vaccines of course are there, produced by E. coli for hepatitis B. That is, it produces surface antigen and it leads to vaccine. It's kind of a vaccine produced by E. coli. So, that is there. So, that is one of the example. Another example is a production of primary metabolites like acids or ethanol. Alcohol. Ethanol is the most common produced fermentation process being known for ages. Nowadays, people use Saccharomyces cerevisiae yeast to produce and ferment the substrate to produce ethanol. In acids, citric acid is produced, glutamic acid is produced. So, glutamic acid is a very good component for the food. It is used in production of monosodium glutamate (MSG), and it is an excellent flavoring agent, which is mainly used in many of the noodle products that you see in the markets. Then you also have glutamic acid, which is also an industrial polymer that it actually normally polymerizes on itself. So, so it produces a glutamates and has different applications in fibers and so on. Microorganisms are also used for secondary metabolite production like antibiotics. For sure, penicillin is a very good example. Streptomycin is another example that is being used to produce this secondary metabolite, and being done by different kind of was essentially done by different kind of was started with penicillin notatum organism, and after mutation it turned into something else and it was being produced. So, that is the secondary metabolite that can be produced. For enzymatic productions, lipases and amylases that are used in lipases and proteases that is being used in the detergents is produced by biochemical methods. Steroids like cortisol is produced by biochemical methods. Then we also exploit the metabolism of microorganisms for microbial leaching, biodegradable waste treatment and so on. So, microbial leaching is essentially you have microorganisms that actually oxidizes iron to iron 3 plus or oxidizes sulfur to sulfate, leading to the production and decrease in the pH values and transformation of the mineral process, and transformation of the process to actually lead to the leaching of the minerals, and basically changing of the minerals, bringing the minerals from the solid forms to the liquid forms, and that is basically is what microbial leaching is. So, microorganisms are used in the treatment of solid and liquid waste. Some of these wastes are not biodegradable, which means that microorganisms cannot degrade them, but many of these wastes are biodegradable, or you need to find the right microorganisms to degrade it. Activated sludge treatment process is one of the treatment processes where microorganisms use your degradable carbon present to produce biomass essentially, and whole of the carbon that is present or majority of the carbon that is present in the activated sludge treatment plant under aerobic conditions is actually transformed to the biomass. So, I can talk a little bit more for leaching and waste is in the leaching. You have iron. So, here iron is in +2 oxidation state, okay, and you have sulphide. Sulphide is also in -2 oxidation state, and basically you have microorganisms that can basically oxidize iron to iron 3 plus, and then this 3 plus, this is solid and this is a liquid. Similarly, you have sulfur as 0, okay. So, here oxidation state is 0, and you have microorganisms that can actually oxidize it to sulphate, and in the process it will also produce H plus. So, this will decrease the pH and lead to leaching of metals. This is one of the way microorganisms can leach. Another way of leaching of microorganisms is not just chemical reaction, but microorganisms MOs can produce molecules called as siderophores, okay, or glutamic acid, the one that we just discussed, and this can basically chelate or complex, can chelate or complex metals and bring those in from the solid to liquid form. So, this is one of the way of transformation of help of microorganisms, where this microorganisms can induce leaching. These microorganisms they can also be used for biological waste treatment, which means that whatever carbon is present or carbon source is present, it is basically is converted into either methane in under anaerobic conditions or biomass and carbon dioxide under aerobic conditions like activated sludge process, okay, which means that you have carbon, okay, which can be called as COD or BOD, okay. This is chemical oxygen demand and this is biological oxygen demand, and basically with the help of microorganisms in under the aerobic process. So, aerobic means presence of oxygen, they can transform this to biomass. So, here this is in liquid, okay, and this change to solid or semi solid. What is its implication? Its implication is you have a water, okay, this water is clean, okay. So, what happens is that you have a chemical oxygen, also you have a carbon material, carbonaceous material. So, maybe I should change this. Okay. So, you have carbonaceous material basically contains carbon, it can be glucose, it can be waste of any other part, okay. There is an oxygen and there is a microorganisms, okay. This is mixed culture. So, what is mixed culture? Mixed culture is several different microorganisms, not just one single microorganism, but several different microorganisms, and once they react, that lead to the production of biomass, okay. Plus this carbon which is in water moves to biomass, okay, transforms to biomass. So, what that means is that your water is clean, okay, and this is what the treatment plant is about. You can also have anaerobic processes where you have carbon material, okay, okay, anaerobic. This is also by mixed culture, and basically this carbon can be converted to biogas. What biogas contains? Primarily methane plus carbon dioxide. So, we want to get rid of as small carbon dioxide as possible and as much methane as possible. Okay. So, under anaerobic condition with mixed culture, it can convert to methane. So, again what happens is that your carbon which was present in liquid, okay, has changed to carbon which is now a gas. So, what does that mean? Your water is clean. Okay. So, that is what means by saying that exploitation of the metabolism. What is the metabolism here? Metabolism is microorganism converting carbonaceous material to biomass or carbonaceous material to methane, and in the process they are getting energy. So, you already would have studied the currency of energy in microorganisms, ATPs, and in this way basically they gain energy to multiply and to survive and do other activities. Next is synthesis of recombinant proteins, therapeutic proteins. Interferon is a very good example that we have actually studied and which we have actually discussed quite a few times, and that can be actually used for production of microorganisms can be used for the production of this recombinant proteins. Now, microorganisms, as you know, would not be only one, right? You have bacteria, you have fungi, you have yeast. Yeast is eukaryotic, bacteria is prokaryotic. So, and different microorganisms produces different things. So, for example, Aspergillus niger, it is used to produce citric acid in large quantities. You have penicillin cryosogenum, it is used to produce penicillin after mutation from penicillin notum. And then you have Aspergillus oreaceae, it is used to produce amylases. These are enzymes that are used for that are used in the detergents, and they are being used for the production of the Aspergillus oreaceae is being used to produce this amylases. Then you have yeast, Saccharomyces cerevisiae, well it is used to produce ethanol, means this is most common organism to for fermentation. So, ethanol just not only for the laboratory use, but these microorganisms are used for production of liquor for drinking purposes, wines, whiskies, beers and so on. So, this is an very well established industrial process, and we already know that this has been activated, or this has been done by the humans for a very long time. Then Saccharomyces cerevisiae via recombinant technology. So, we modify the genome of Saccharomyces cerevisiae and can be used for production of hepatitis B surface antigen. So, this is for vaccination purposes. Then you have E. coli. So, then you have the bacteria, these microorganisms that are used for production of different things. So, streptomycin is produced by Streptomyces griseus. Then you have Corynebacterium glutamaceum is used to produce L-lysine. This is an amino acid that is used. It is also used, this microorganisms is also used to produce glutamic acid. Then you have Propionibacterium shermani, that is used to produce vitamin B12. Then you have Leuconstock microorganism that is used to produce dextron. You have E. coli that is used to produce insulin, growth hormones, interferons, and I have also said that it is used for production of hepatitis B surface antigen, but the method has now moved to yeast Saccharomyces cerevisiae. Then you have alkalogenes eutrophorus, this is used for production of poly 3 hydroxybutyrate. This microorganism alkalogenase is also used to produce siderophores alkalogen, but only at a laboratory level at the moment, and research is going on to actually see if this can be used to produce siderophore in a much larger quantity, especially the tetradentate ones. So, the next part of the lecture is the biosafety issues in the microbial technology, and it is quite an important actually, and uh, and we need to learn how to actually address the biosafety issue in the microbial technology. And the reason is that there is a potential of genetically modified organisms or recombinant strain to infect humans, plants, animals to cause disease. So, as you know, as of now, in the last lecture we talked only 10 raised to power minus 22% of total estimated microorganisms whose genome we have sequenced. So, we really know very small amount of the microorganisms that are present in the environment with us. So, what happens when a genetically modified organism or recombinant strain is leaked out of the laboratory? Once it is leaked out of the laboratory, we cannot predict how it is going to interact with its surrounding, whether it will mutate again, what it will do, and how it will affect the natural balance that is already present. So, will it outcompete all the native species and become a predatory species where it is the only one that is available there, and that might lead to a lot of challenges, even ecological challenges, where the fine balance in the nature would be disturbed, and this actually creates a issue on the safety side, and that is why as I said before, you can use a genetically modified organism or recombinant strain to produce a bioproduct, but you will not use them in environmental processes or microbial leaching processes or wastewater treatment, because what happens is that anything that comes into the contact with this GMO organism, the 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 things that have come in contact, for example solid substrate or the water or anything, has to be autoclaved. That means everything has to be killed before discharge, and that is the rule of thumb. All the environmental protection agency everywhere, this is rule across the world. So, and there is a very good reason for that, because if we cannot predict what will happen to this organism in the environment, then we are playing, we do not know how nasty or how effective or what they will do, and this is a far higher risk than any other benefit that comes along with it. Next is that when we use these microorganisms for production of a product that is for human consumption or for humans to come in touch with them, then you also have to make sure that the toxicity and the allergy, when you are using this microbially produced biochemical, this should not be there, because if that is there, then it defeats the purpose. That means the product quality is not there. So, this is another thing that you have to take care of when you are using a recombinant strain. With natural strain, this is not that much of a problem, because naturally they have been selected in a way to do a particular job, and because of the selection pressure of the microorganisms of the environment, selection pressure of the environment actually, not microorganism, has ensured that the recombinant molecules that are being the molecules or the proteins that are produced by natural microorganisms are effective and are what we are expecting, to do the job that they are supposed to do. However, when you do genetic engineering recombinant, you are not sure exactly how good was your selection pressure, and whether whatever you have tinkered with would not lead to toxicity or allergy, and hence this is important to test this before we put this products in the market. Then other medically relevant implications like increasing the environmental pool of antibiotic resistance microorganisms or transfer antibiotic resistance gene. This is very important. So, that is why you do not really want to discharge microorganisms which are genetically engineered without autoclaving, and even to an extent disrupting or damaging completely their DNA and destroying everything before discharging of the wastewater containing it. Because for example, we in the laboratories have organisms, genetically modified organism with a to make sure that they are the ones that get selected to have an antibiotic resistance gene in them. And now if we release these microorganisms in the environment, from the product point of view they are fine, they do not do anything, but from the antibiotic resistance point of view they are already resistant to certain antibiotics, and this antibiotics would be are this antibiotic gene is available for other microorganisms, other native natural microorganisms to pick up, and when they pick it up, basically this microorganisms are unaffected by the antibiotic. And imagine if these microorganisms are human pathogens, then basically if it's in fact a human, the antibiotic that this person is going to get for the treatment of this microorganism or for the treatment of this bacterial infection will not work. So, this is a very big problem. Actually, it is a very serious problem, considering that antibiotic resistance microorganisms and genes are getting prevalent in the in waters across the world, and thus it is important really to take care of this problem. But from biotechnology point of view, microbial technology point of view, our aim is essentially never to discharge any genetically modified organism or recombinant strain into the environment without autoclaving, without pre-treatments, and that is why all the industries and everywhere in the labs follow these procedures. Then another issue that can be is that if you use microorganisms to produce your biomass, produce your bioproduct, at the product is out, it is not in the biomass, and you have a lot of biomass with it. So, you have a problem associated with disposal of the spent biomass, how to actually get rid of that biomass. So, of course, as I said, one way, if it is genetically modified or it is a recombinant, you can directly go for autoclaving, destroying the biomass. But if it is non-genetically or native, then this biomass are also, for example, in the wastewater treatment plant, you produce a lot of in the activated treatment processes, you produce a lot of biomass, a lot of sludge, and then this sludge is basically are used for anaerobic digestion. So, where it is not the water treatment, but anaerobically this biomass is degraded to produce methane, and whatever is left of that biomass, if it is not, if it does not contain a lot of metals, then it can be used as a fertilizer as well, and this is a normal process, but that is only valid for microorganisms that are non-genetically modified. So, basically microorganisms which are native or natural. Then you have to also take care in the microbial technologies safety aspects associated with contamination, infection or mutation of the process strain. So, if you have a strain that gets contaminated with something else, or if it is highly contaminated that it contaminates something else that you are also working on, so it is not very good. Also, whether it is the strain is safe to work with, whether whether it will mutate to more dangerous strain. So, that is always a challenge, and basically we work with microorganisms in microbial technology, product formation, product like ethanol production or antibiotic production or interferon production. We only work with the strains which are has a very good safety record, which we know very well their genomic code, which are generally considered as safe and which do not infect humans or other mammals. So, we only work with those strain, we do not work with any strains, or normally we will not work with any food based product or any human consumption product with a strain that is infectious, even if its productivity is thousands or 100,000 times more, we will not work with that. So, this is an concern, and this is an important point to consider while selecting the strain as well. So, as I said, when you have that kind of things, you only work with generally regarded as safe category microorganisms. So, which microorganisms you can work with? You can work with naturally occurring microorganisms by selection, things like that. If you want to work with genetically modified organism, you have to work with only microorganisms which are considered as safe. So, E. coli for example is considered very safe, Saccharomyces cerevisiae is very safe, Pichia pastoris is very safe. So, this 3 microorganisms for example is considered very safe, and all the genetic engineering that people does normally does in this microorganism. Nowadays Bacillus subtilis, people are also working on that, it is also considered as relatively safe and people are working on that. So, this is the kind of microorganisms that you will work with, and everywhere all the institution have expert committee that review on case to case basis the biosafety features. So, they will, you can work on genetically modified organism only when an expert committee allows you to do it, otherwise you will not be allowed to do it, and they review your application on case to case basis, that what kind of safety levels are needed, whether those safety levels are in place, what kind of application we are looking for. So, of course, if you are working in a field of biomedical, you might be working with infectious microorganisms, but then the safety level requirements are very high, and the products that you are producing will not come into the contact with humans, and maybe you will never produce any product with them. So, for the human consumption product, you work with, as I said, GRAs, generally regarded as safe category microorganisms. So, having said that, there are issues with microbial technologies with safety and safety and things like this. So, it also becomes very imperative that what are the good microbiological practices that are there. So, first thing is that you have different safety levels as well, but even the normal microbiology lab, it is not a chemical lab, it is a microbiology lab. So, you are working with living organisms that has a propensity to infect, to contaminate, to move around. So, what are the general good microbiological practices? The first thing is that person must wash their hands with soap, germicidal soap, after handling viable microorganisms and even before handling as well. So, whenever you work with a pure culture, you would work in a hood and you would disinfect the hood before and after use. You would disinfect with ethanol, 80% ethanol, 70% ethanol, and you will use UV rays in the hood itself to basically disinfect. And after you have worked with it, before you have worked with it, you will sanitize your hands with soap and then with ethanol, and after you have worked with it, again you will sanitize your hands with soap and then ethanol. So, this is a good practice always when you do it, because if you do not do it, then you cannot be sure whether the microorganisms that you are working with is will remain pure, because it may get contaminated. That is first part. From the microorganism point of view, from your point of view, if you do not wash your hands, then you have a viable microorganisms on your hand which is maybe genetically modified and not suitable. It is not good for you to keep it in your hands and transmit it to others, also to yourself. Next point is that eating, drinking, smoking definitely not permitted in the working area, not permitted in any lab, because again you have viable microorganisms, and what you are eating also contains are not completely sanitized things. So, that is definitely not a place in labs, are not a place, or the working areas are not a place to actually do these activities. Food that is to be stored should be outside the work area in cabinets refrigerated designated for this purposes only. Is the same reason that food also have contamination. So, you do not want cross contamination from food to your working place or working culture, and the other way around. So, this is definitely one of the very important thing considering when you are working in microbiological labs. The next part is mouth pipetting is strictly prohibited. Again, you should definitely never pipette with mouth. In some of the chemistry lab earlier that was used to be done, but here absolutely not, because you are again working with live microorganisms, you increases chances of cross contamination. So, all the procedures are carried out in a way so as to minimize splashes and generation of aerosols. Again, if you have splashes or if you generate aerosols, you transfer, you increase the risk of contamination. After the work with viable microorganisms, workspaces are decontaminated thoroughly. So, this is I already explained to you all how to basically decontaminate it. We decontaminate it with ethanol, we decontaminate it with UV lights in the area, in the hoods where we work. And this is important because you might be working with viable microorganism 1 and your colleague might be working with viable microorganism 2, and you do not want microorganism 1 to infect or contaminate microorganism 2 or the other way around, as well as you do not want yourself to be contaminated with those two microorganisms. So, that is very important. Of course, you never wear um, your daily clothes. You always should have laboratory coats, aprons. You should wear full shoes, never floaters or sleepers in the lab. These are general good practices, not only for microbiology labs but also for in generally. And all cultures, stocks and other waste are decontaminated and autoclaved before disposal. So you basically, let's say you have a spent media in which microorganisms were there, you collect them in one of the flask, you basically put that in the autoclave, so that is done. Let's say now you have only small amount of flask which had culture, but you actually removed everything, still it has enough amount of microorganisms to basically get discharged. So you basically bleach it, you add bleaching agents to basically remove whatever is left over, and you do not discard anything without autoclaving. So even if you have petri dishes, they go only out of the lab after autoclaving. Without that, you increase the risk of contamination in the environment, and once these things propagate in the environment, there is no way to actually control it. So I cannot stress enough on the need for controlling these things, and always using autoclaves, decontaminated good microbiological practices, uh, so that anything that we do in the labs or in the process industry does not come out. So most of the time, from the industrial point of view, most of the time industries work on zero discharge process. Basically they have no discharge at all. So whatever water they whatever wastewater they generate, they treat that wastewater and they recirculate that wastewater and use it. So that is how a good microbiological practices are, and that is how we should all follow it. So with that, I would like to end this unit. Um, so just to give you a recap, for all the lectures that we had, we went through we looked at the what are the things that microbiology can produce, why biotechnology is important, what kind of products it can produce, why it is the only technology that can actually replace chemical industry possibly in future or maybe in some distant future, but it is definitely going in that direction. Why cost is important, what kind of substrates are there, are what are shakers, shake flask, what are reactors, how are the big reactors operated, what are the scale up principles are there, how the microorganisms follow the monod kinetics, how can you look at the growth rates of the microorganisms, how can you determine the growth rate of the microorganisms. Then we also looked at the downstream processing, cell disruption, filtrations, centrifugations, techniques such as adsorption, chromatographic techniques, precipitation techniques, and all these kinds of things that are there. Then we look at the strain preservation, but before that, strain collection, that how you can go into the natural environment and collect the strains that are needed for your work, how you can mutate those strains using chemicals or physical methods or genetic engineering, how metagenomics can be used. And then we had a look at how to preserve those strains, why the preservation of strain is important, what are the centers, cell culture sections which are there. Then we had a look at what are the products that they can actually produce, then what are the safety features that are needed, what are the good microbiological practices that are there, and all these things actually leads to the production of microbial technologies and use of microbial technologies for production of the products in a cost effective way, which is also green to the environment. So with that, I thank you very much, and I hope you enjoyed this lectures. Thank you.